Hey, so this is our last section about the cells, and for this we're going to talk about enzymes. Enzymes are really important because they are protein molecules that help make it easier for other chemical reactions to happen. So they speed up a chemical reaction, that's what they're important for in chemistry. And the enzyme itself isn't part of the reaction, so it doesn't get used up. It can go and help another reaction, and then another one, and another one. The enzymes make the metabolism in your body work. They're the things that make the reactions inside your body work. In biology, we don't so much need enzymes to speed up reaction. A lot of times we need enzymes to make a reaction possible. A lot of times there just isn't enough energy in an organism to make a reaction happen without the presence of an enzyme. So enzymes become vitally important in proper physiological functioning. One note, enzymes are always proteins. Remember back from our chemistry chapters, proteins are strands of amino acids. Okay, all enzymes are proteins. Not all proteins are enzymes, but all enzymes are proteins. All right, so this is a graph that shows time on the x-axis, the amount of energy on the y-axis. And here we have a reactant, which is a big molecule. And we're going to break it into two smaller molecules, which we call the products. In order for this reaction to get started, we need to add some energy, and that's called the activation energy. So even though when we get to the end, we will have released energy, and there will be less energy in these two molecules than there were in the whole one when we started, because we're going to break this bond. We have to add energy to get it started, and that's called the activation energy. Okay, think of the activation energy as the friction that you use to strike a match. Okay, so you strike a match, that friction provides energy that starts a chemical reaction in the chemicals on the tip of the match, and then from there the fire continues to burn without you having to add any energy to it. Okay? It's that activation energy that's required to get it started. With an enzyme lowers the amount of energy that is required to get a reaction going. So in chemistry, this is important because you can have reactions happen with less energy in the whole system, with less energy being used. In biology, without an enzyme, most of the time, we can never get this much activation energy into the system, so the reaction never happens without the enzyme present. And in fact, there are a number of diseases or conditions that result from a person lacking an enzyme. We'll come back and talk about some of those in a little bit. So each enzyme recognizes a specific molecule or molecules, and that's called the substrate. Okay, substrate is a term we use for just like wherever or on whatever something happens. Okay, plants grow on a substrate. That might be dirt, sometimes it's a rock, sometimes it's another tree. Okay, enzymes recognize a specific molecule, that's a substrate. The substrate is going to bind to a specific site on the enzyme, and that's called the active site or the binding site. The binding site and the whole protein molecule, remember that an enzyme is protein, whole protein molecule is going to change shape when the substrate binds to it. And that's called the induced fit model. The binding of the substrate to the enzyme changes the shape of the enzyme, and that, the physical changing shape, makes the reaction easier to happen. Now think of, here's an enzyme, okay? This is the enzyme lactase. One note about chemical names. If you see something that ends in ASE, it's always an enzyme. Now, not all enzymes have names that end in ASE, because that would be too simple, wouldn't it? However, anything that ends in ASE is an enzyme. Now, lactase is the enzyme that breaks down the molecule lactose. Anything that ends in OSE is a sugar. Glucose, fructose, lactose, galactose, all sugars. So lactase breaks down lactose. So here we have the active site available. Our substrate is lactose. Lactose binds to the enzyme. The enzyme changes shape. As that happens, water gets added, and the water gets split between the two resulting monosaccharides. The changing shape of the lactase enzyme helps to break this chemical bond, and galactose and glucose are released. So lactose is a disaccharide, you remember that from our previous lecture, two sugar monomers, and those sugar monomers are glucose and galactose. Okay, so with the help of lactase, lactose is broken down into glucose and galactose. Okay, this is a hydrolysis reaction. Let me see if I can write hydrolysis so you can read it. Okay, hydrolysis. Hydro meaning water, lysis meaning to cut. So it literally cuts water. If you put these two together, glucose and galactose, and you're going to remove water to create this chemical bond. That's a dehydration reaction because we take the water out. Cool, right? Okay, the other thing that's important is that this enzyme hasn't been changed at all by helping this reaction happen. So it's ready to bind to another lactose. Another lactose comes along, binds, the lactase changes shape, breaks the bond, releases the products, gets ready for another substrate. Okay? Now, we don't always want our enzymes to be working. If there's no lactose in your body, you don't need to be making lactase. So, biological systems and all organisms have enzyme inhibitors, which is something that binds to the enzyme and prevents it from binding to the substrate. So remember, each enzyme is really, really specific for what it binds to. And whenever a protein binds to something, it changes shape. So here's one form of enzyme inhibition. Here is the substrate and the active site. Okay, substrate can bind, something happens. Now, if we bind something at this site over here, or this site here, either one is going to change the shape of the protein and prevent the substrate from binding. So this inhibitor is going to bind at the active site, and it just blocks the substrate. The substrate can't bind because something else is blocking the seat. Somebody stole its seat, okay? Now this one blocks at a different site, but remember, when you bind something to a protein, it changes shape. So by binding over here, changes the shape of the active site, and now the substrate can't bind. Okay. Penicillin is actually an enzyme inhibitor. It blocks an enzyme that bacteria use to make their cell walls. So if the bacteria can't make cell walls when they reproduce, then they can't reproduce, the bacterial cells die, and your bacterial infection goes away. That's why penicillin is such a good antibiotic. Some cancer drugs block enzymes that promote cell division, mitosis, and help cells to make more of themselves. So by blocking that, they prevent the growth of the cancer. Ibuprofen, or Advil, inhibits an enzyme that sends pain signals through your neurons. Without that signal, your neurons don't send the signal to say, ow, that hurts. Okay, so that's enzymes. I'm going to post a link to another video that's going to help you understand this a little more if you're not quite clear on enzymes yet. But that's the end of our cell chapter.